Hey everyone, this is another short video to help you prepare for your first practical exam, and this is going to cover the integumentary system. So let's start by just reminding ourselves that there are 4 main tissue types that you are responsible for knowing all about. And in your lab manual, there is this pretty little flow chart over here. I really recommend that you spend some time with it and honestly just commit it to memory. Because when you organize information in your mind, it makes it so much easier to pull that information out later and to understand the relationships between them. So we are mostly focused on connective tissue and epithelial tissue in this portion of our lab course, but you want to be aware of muscle and nervous as well. So within connective tissue, we are looking predominantly at connective tissue proper, and we're going to focus on areolar, adipose, and maybe be aware of dense regular and its location. And then we are definitely interested in all of the epithelial tissues, both simple and stratified, and then the different shapes, squamous, cuboidal, columnar, and pseudostratified, with an emphasis on the squamous and cuboidal tissues. And also for stratified, we're very interested in the stratified squamous, keratinized, and non-keratinized. So those are the areas I'm going to focus on in this video. For each of the 4 main tissue types, you really need to have a sense of their basic structure. So what are the cells like? What shape are they? You want to know, do they have an extracellular matrix? And if so, what's in it? You want to know about the basic functions. What do those tissues do? If they have any unique characteristics, you want to be able to list those. And you definitely want to be able to identify places in the body where you find each of those tissues, and that usually relates to their function, and you want to make sure. So for example, one of the functions of epithelial tissue is that it can be protective, and we also find it lining cavities. Well, maybe those 2 things have a relationship. So you want to think about those. So study the charts in your lab manual on epithelial and connective tissue, because they ask you to identify some of the different functions and some of the different locations. So that should really highlight those ideas for you. Okay, let's dig into the microscope work, or the histology. One of the things that's important when you are looking at cells under the microscope is to remember that depending on the plane of section, they might look very different. So these 2 images are showing us simple squamous epithelial cells, so 1 layer of squamous cells. If we look down on them, we can see they have a big round nucleus, and we can see that kind of irregular shape of the cells. I think the word is, I can't come up with the word polyagonal. I don't think that's the right word. Someone help me and let me know via email what that word pronunciation is. But we can see the irregular shape of these cells. But over here, we also have simple squamous cells lining the glomerulus of a kidney. But notice they look super thin, and the nucleus doesn't look round, it looks flattened. That's because we're looking at these cells from a different angle than we're looking at these. So we're looking at these ones sort of from a top view, like a top-down view or a superior perspective, whereas these ones we're looking at from the side, so we really see how thin they are. By the way, this is another great example of how the location and characteristics and function go together. These are very thin cells. They are lining this space right here, and they're really good at allowing things to either move across because they're so thin, like diffusion. We'd see that in the lung tissue, for example. Or in this case, they may play a really important role in filtration, which is similar but not identical. But that thin structure to them is essential to their function. Okay, here we have a beautiful example of cuboidal cells lining this duct right here. So we can see the big round nuclei and the sort of boxy cell shape around them. I mean, the nuclei are almost as big as the cells. So these are great examples of simple cuboidal epithelium. These are in the kidney as well, just like the previous slide showing the glomerulus with the simple squamous epithelium. Now, if we look at stratified squamous epithelium, we have two kinds. So we're going to have stratified non-keratinized squamous epithelium. We can see the nuclei all the way to the apical surface. And then we have stratified squamous keratinized, where there's no nuclei up towards the apical surface. So take note, these are both stratified squamous epithelia, so they're going to have multiple layers. They're both going to have an apical and a basal surface, but this is going to be skin, like in the integumentary system, and this is going to be more likely to be found, say, in the esophagus or vaginal canal. One of the things you should notice is that we have over here a very distinctive layer where we can see cells that have granules in them. That would be stratum granulosum, and that means above it we would expect to have stratum corneum. We might have a stratum lucidum in between those two layers if it is thick skin, and below we'd have stratum spinosum, and then the very bottom layer would be stratum basale. And we can see those same layers over here as well. We're just not going to have that keratinization in stratum corneum up here. So similar, but a little bit different. Also, take notice, we have an underlying dermis. We always find that with epithelia because remember, one of the characteristics of epithelia is that they are avascular, but dermis has a connective tissue in the dermis underneath has a great blood supply. And then also remember that we name the epithelium for the uppermost layers near the apical surface. So these are the squamous cells that we are naming this tissue for. If we come down deeper, we can see that actually some of these cells are not squamous. They are more irregular shaped, leaning towards cuboidal-ish or even columnar down the bottom. But we don't name our tissues for the deep layers. We name it for these surface layers up at the top. In visible body, you can compare thick skin to thin skin, and as a result, we'll be able to see all of the layers. So you could try naming the layers. You could pause this video and see if you can identify the layers, and then I will just pop right over to the next one, and here are all of our layers. And so we can see our dermis underneath, and our stratum basale, spinosum, granulosum, there's lucidum, and then corneum at the top. And we notice that it's peeling in both. That's pretty common for keratinized epithelium. It's an artifact of preparing tissue for microscope slides, and so you get that peeliness that you don't tend to see with non-keratinized epithelium. And you could also talk about how these are dermal papillae. So you might be able to, you could talk about what kind of connective tissue, loose areolar, might we find in the papillary layer of the dermis. But we can also dig into those layers in another model in a second. Okay, here is yet another example of our epidermal layers. This is also from visible body. So you can, read the little snippets over on the left in visible body. And here's a beautiful example of stratum granulosum. And then if you can find stratum granulosum, you know that the deeper layer is going to be spinosum. Above it would be lucidum or lucidum, and then above that would be corneum, and the bottom layer would be basale. So now we also looked at connective tissue under the microscope, and we looked at loose areolar connective tissue. Both of these images are loose areolar connective tissue under different stains. So you might notice they look a little different at first, but you'll notice there's the fibroblast nuclei, and notice that the cells are really spread out in connective tissue, unlike in epithelial tissue where the cells are really packed together. Notice the big, thick collagen fibers here, and then also here and here, and then you see the very fine sort of black elastic fibers in both of them. Those are common characteristics of loose areolar connective tissue that you would want to be able to identify. You also looked at adipose tissue. This is another image from visible body, and you can see the really beautiful lipid droplets inside each adipocyte or fat cell, and it doesn't pick up stain very well, so you get this kind of white, circly, bubbly looking tissue. You should also be able to spot the nuclei sort of pushed off to the side by the lipid droplet. And then the last thing in the integumentary system that you want to spend some time with is these models of the skin. There is a great one in Visible Body, and you can rotate it and you can expand it and you can click on all of the structures to find out what they are and gain more information about each one. You can study the layers of the epidermis and the dermis and the structures within them and then the hypodermis. But don't forget, and I encourage you to do that. But don't forget, you also have the models that were in the lab of the skin. You want to be familiar with those models. And you also have the diagrams in your lab manual that you want to be familiar with. So you need to be able to recognize and identify these structures and cells in all the different kinds of models that might show up on your practical. All right, good luck studying.